Hello again and welcome to part 2 of this 8-part series in my beginner's guide to colour genetics in budgerigars. If you haven't seen part 1, then I will put a link on the top of the screen now so you can go across and watch part 1 where we covered many of the main terms. In part 2 we're actually going to make our very first prediction, so we're going to get into actually predicting an outcome of pairing two budgerigars. And in doing that, we'll begin to understand the relationship between dominant and the dominant gene and the recessive gene. So that's what we've got to look forward to in part 1 of my introduction to genetics. As always, any of the information that's contained in these videos is reproduced and in a bit more depth in a PDF book that I've produced that runs alongside this series. If you want to download a copy of that PDF, it is free of charge and it's available across on the Shed Talk Facebook page. So please do go across and visit the Facebook page, join it, and you'll be able to download that free of charge. If you do enjoy the series or any of the other Shed Talk videos, then please do hit the like button. Don't forget to subscribe and hit the bell if you want early notifications. So, of course, in part 1 we covered a number of key areas. So from part 1 you should have a reasonable understanding now of what we mean by the cell, the chromosomes and the genes that lay within those chromosomes. You should understand the term locus and the allele. Also understand what we mean by when a bird is considered homozygous for a particular gene and heterozygous for a particular gene. So that was all covered in part 1 of this series. Well, before we begin to discuss predicting recessive and dominant genes, I just wanted to mention someone that's often considered to be the father of genetics, and that's Gregor Mendel. He did loads of work over about a decade in the 1850s to the 1860s, where he studied the interaction between various genes, and he used well, one of the things that he used was the pea plant, and he looked at colour of flowers and the various shapes of the pea pods, and from that he made a number of I suppose theories about how those interactions happened, and he developed some general rules and principles, and we still today refer to those principles, and they still hold true today when we're considering the colour genetics in budgerigars. Those rules you'll often hear people refer to as Mendelian laws, so the Mendelian laws or Mendelian rules of genetics. And by and large, those are the rules that this series and indeed my PDF book follows. So there's more details about how, or the work that Mendel did in the PDF document. So do across, if you want any more information on that, please do go across and look at the PDF document. Always worth a read. Father of genetics, bearing in mind he was doing this without any of the modern facilities we have today, like microscopes and all that sort of thing. He was using pure mathematics, I suppose, to work this out, and that work still holds true today, as I say. So what I want to do now is to introduce you to one of the main tools that we will be using throughout this series in order to make predictions. There are a number of other tools that can be used, and you may have come across some of those, but this will be the main tool that I will be using. It's my preferred method of making a prediction, and that's using a tool called the Punnet Square. We'll nip over to my main computer now, and I'll bring one of those up and we'll begin to have a look, and we will describe what the Punnet Square is and the various elements of it, and how we would use that. So here's an example of a Punnet Square that I've produced on, it's actually on Google Sheets. You could do something similar in Excel. So I will just explain the various elements to it. So before we move on to using the tool in detail a bit later on. So first of all, I've put across the top up here, hoping you can see that, the mating, or the pair that I'm mating. So here we've got a sky blue cock crossed with a light green hen. But this is the main bulk of the Punnet Square, is the bit below it. So, and you can see that I've replicated the pairing over this side here. I just click on it. You can see the sky blue cockbird, and on the left hand side I've got the light green hen. I've just coloured it for ease so you can see it. So that's the pairing. It is I think fairly common that you put the cockbird across the top and the hen on the left hand side. I think that's a common way of doing that, so I'm just following that convention. And then the bit directly underneath here, where we can see you've got underneath the cockbird, you've got BL1 and BL1. Don't worry about what those mean for the time being, but those are the genes that we're going to be or that we're going to be investigating, and those are the pair of that gene that cockbird has. Remember, in the first video we said that each bird will have a pair of a particular gene, and for the light green hen we've got the B there and the B there. So each one of those represents an allele of a particular gene, and in this case both of the allele for this gene in each of the birds are the same, and therefore are homozygous for that particular gene. So that's the Punnet Square. The bit down here, so the 4 squares that I'm now clicking on, is where we will place the outcome, and we're actually going to do this one in a short while, so you'll see how that particularly works. So there we go, that is the Punnet Square and how we'll use it, and we'll continue to look at that in more detail as we run through this particular video. What I'd like to do though, before we start talking about recessive and dominant genes, is to talk a little bit about the green and blue series of budgerigar, because that's what we're going to be looking at. So we're going to look at the gene reaction between the green budgerigar and a blue budgerigar, and it will help us, if we understand the difference between the blue series and a green series budgerigar, then it will help us understand some of the other varieties and how those varieties reflect that series, so blue and green series. So in general, that we consider budgerigars to either fit into one of those two series. That's our general view. So it can either be a green series or it can be a blue series. I'll put on the screen now it's a video of a blue and a green series budgerigars, and we'll discuss the difference. I suppose to begin with, you'd probably say there's an obvious difference, and that's that one of those birds is blue, has a blue body, and one of them has a green body. But however, if we look closely, what is actually happening here is that the areas of the budgerigar that are yellow in the green series bird are white in the blue series bird. So if we look at the face of a blue bird, of a blue series bird, it has a white face. The markings on the wings reflect that also, so they are white rather than yellow that we see in the green series bird. So the green series bird has a yellow face and yellow markings on its wings. So if we then reflect that in terms of the body colour, you know, if we were to take the if we added a blue pigment to the blue parts of the budgerigar, we end up with green. If you mix blue and yellow together, we end up with green. So in effect, the blue budgerigar is just a green budgerigar minus the yellow pigment. Um, so that's how those two work. So if you remember that, so a blue budgerigar is actually just a yellow bird without the yellow pigment. So as I say, all those bits that are that would appear yellow in the green series bird appear white in a blue series bird. I will very quickly mention at the end of this that whilst we consider birds to be broken down as being either blue series or green series birds, when we're talking about a yellow face bird, it becomes slightly more complicated. But we'll come on to that in the part 8 of this video, and we'll discuss yellow face in a little bit more detail. So there you go. It's not just about green and blue. It's actually more about yellow and white. So there we go. That's the green and the blue series. You may know this, that originally all the budgerigars were green series, so that's considered to be the wild type, and the blue is the mutation. So let's talk a bit about the blue mutation now. So here we are back over at the main computer looking at the Punnet Square that we looked at earlier, the same sky blue cock and light green hen. Before we go on to look at how we use this in order to make the prediction and then talk a little bit about dominant and recessive, I want to talk a bit about the naming of genes. So when a mutation occurs, the mutation only tells us really what the impact of that mutation. So in the case of the mutated wild gene in the green bird that produced the blue bird, it's referred to as the blue gene because that's the impact of that mutated gene. What we didn't know, what it doesn't actually tell us in truth of fact, is that gene produces a green bird, because it might have been more than one gene that produced it, and just a single gene change in NA prevented it from producing the green bird, and you ended up with a blue series or a white series bird, blue series bird. So we name the gene after what the visual impact of that of the mutated gene. So in this case, it's referred to as the blue gene. You'll notice here that I've I've put some just single letters or a couple of letters and a number in to refer to the allele of the particular bird. And so if we look down the left hand side first for the light green bird, so if I click on these here, you'll see that those two are marked with an uppercase B, and the ones across the top are lowercase and are BL1 and or both BL1. So let's just talk now a little bit about the second convention then, and that is how we represent the blue gene on the Punnet Square. And we use we would normally just use a number of letters to represent that, and sometimes we use letters and numbers, as in this case. The other thing that we're going to do in this particular series, it's not used all the time. There are a number of conventions around how you then represent those in the Punnet Square. We will always, in this video, we will always use where a gene is the dominant gene, it will be in uppercase, and where it's a recessive gene, it will be done in lowercase. So you can see here that the unmutated wild type gene, the B, represented here, is in an uppercase, so a capital B. That means that's the dominant allele, and the sky blue, which contains the two mutated allele, BL1, is recessive. So dominant is in a capital, recessive tends to be placed in lowercase. The reason it's BL1 you see here is because the blue gene is known to have mutated or this particular gene is known to have mutated more than once, and therefore we needed more than one representation for it. So in this case it's BL1, and we'll talk a bit about multiple allele in a future video. So that's the various conventions dealt with then. So how do we use the Punnet square? Well, it's relatively simple. You'll notice that we've split the two allele into individual squares, and we can consider that in effect to be the single sex cell that's going to be moved over. So in here it's the sperm, and you'll remember that in the case of the sex cell it can only contain one of the allele, and the egg will only contain one of the alleles. So there's the various ones it can pass on. The blue cockbird can either pass on, it can only pass on BL1 and BL1 because that's all that it contains, and the light green hen can only pass on the B and the B. So all we're going to do is to bring these down, add them together. So we take the BL1 from the blue cockbird, and we then add the B from the light green hen, and that will give us a new bird. So that's the merging of the sperm with the egg in the two birds, and it gives us a new bird that now contains two allele that are different. So it's heterozygous to the blue gene. It contains one BL1 gene and one B gene, and in fact all of these cells will be the same because that's all each of them can pass on. So they all contain BL1 and B. So I'll just fill out the rest of these. So the big question then is, what will the bird look like? Because it's containing different genes to both its father, which can just contain two BL1s, and different genes to its mother, or sorry, not different genes, different allele to its mother, which just contained the B. We said that the uppercase B is dominant over the lowercase BL1, and therefore this bird will look like the mother, so it will be a green bird. This is because the wild type original gene that allowed the production of, in this case the production of the yellow pigment to produce a green bird, is dominant over the mutated gene, and therefore it will always produce a green bird. So so that's the way we use the Punnet square, and we're beginning to describe the way that dominant and recessive interact. So let's have a talk about that now. Well, we will return to the Punnet square in just a moment, but before that I just wanted to talk briefly about the dominant recessive then. So I think this is the easiest one really of all of the interactions between alleles that there is. So in effect, dominant merely means that if the bird contains at least one of the alleles, is a dominant variety, then it will always look like that variety. So in the case of this, the one we've just looked at, so providing the bird contains one of the dominant non-mutated blue allele, the bird will always appear green. In order for the recessive trait to show up in a bird, then both alleles must be for that recessive allele. So both of them must be. So in the case of this latest one, then they both must contain the mutated blue allele. So that's quite simple then. If it contains one of the dominant alleles, then it will always look like it. And for recessive, it must be homozygous for the recessive gene. What I'd like to do now is to just go back over and have another look at the Punnet Square, and we'll practice a little bit by looking to cross two birds that contain only one of the mutated blue allele, and see if we can predict the outcome of those birds and see what that would look like. So here is the Punnet Square then for the green split blue cock across a green split blue hen. Exactly the same as before. So we've got the two allele for the cock bird across the top, and the two allele for the hen on the left hand side. So if you want to have a go at completing this yourself, then please do so. Just pause the video now and have a go at completing it. Well, I hope you managed to complete it. What I will do now is complete it and you can check your answer with me. So remember that we bring the top down and the left across for each of these squares, so we end up with a bird with the new bird with one of the allele from the cock bird and one allele from the hen. So this one here would quite clearly be a B, comma B. This one here we bring the top down BL1 and comma bring the B across from the hen, and down here we bring the BL1 across from the hen, we bring the B down from the cock bird, and then the BL1 from the hen, and here we bring the BL1 down from the cock bird and BL1 down across from the hen. So we end up then with three different possible outcomes from this crossing. We've got the BB here, which in effect is a green bird, the B1 Bs, these two, which will be, remember that the B is dominant over the B1, so that would be a green split blue bird, and then the B1 and B1, which are both the recessive allele. And if you remember, if a bird is homozygous for the recessive gene, then it will be of that recessive variety. So this would be a blue bird. So that's the outcome from there. And what I want to do now is to talk a little bit about some of the things we've identified from this crossing. So we'll go back across and have a look at that now. Okay, so one of the things that last prediction showed us is that actually with the dominant recessive, you can have a bird that looks identical but has different alleles for that gene. So we had the bird, the green bird, that contained both of the non-mutated allele, and we also have a green bird that contains one mutated allele, so one mutation for the recessive mutation, and one that was of the non-mutated gene. So first of all, I'll talk a little bit about that, where we've got a dominant gene and a recessive gene in the same bird. Okay, that's normally referred to as the bird being split for something. So in this case, we've got a green bird that is split for blue. So in order to understand when someone says it's split for something, all that means is that it contains the recessive allele for that particular variety. So green split blue just means that the bird contains one of the recessive blue allele genes on it, or one of the genes is the recessive blue type allele. The other thing is that it means that we've got birds that look the same but have a different genetic makeup, and we refer to the genetic makeup of a bird as its genotype, and we refer to the way the bird looks as its phenotype. In actual fact, phenotype means much more than that, but it actually means the way the bird behaves and all the rest of it. So its outward appearance, I suppose, and characteristics. But from the point of view, in most cases, from the point of view of bird breeders, when they're referring to a bird's phenotype, they're referring to the outward appearance. So with dominant and recessive varieties, we will have 3 genotypes, but only 2 phenotypes. So that's the difference between the pheno and genotypes on a bird. Well, I hope you enjoyed that quick run through of the recessive and dominant interaction of allele on a particular bird. Like I say, if you want any more information or go into a bit more depth, then do go over and have a look at the PDF. By now you should have a full understanding of what we mean by recessive and dominant and how those 2 allele will interact. You should also understand now what we would mean by a bird that is split for a particular variety, and remember it can only be split for a recessive gene. And then finally, we would be able to understand what's meant by a bird's phenotype as compared to its genotype. In the next video then, we will cover incomplete dominance, which is another type of reaction between the various mutated allele. But until then, please do stay safe and enjoy your birds.